Hello, today I will be explaining how DNA is put together, how it copies itself, how the processes of transcription and translation occur, and how different types of mutations occur. I will be using a diagram of a DNA strand that is 10 nucleotides long. This DNA is a double-stranded helix, which is how DNA replicates itself. Nucleotides form the basic structural unit of nucleic acids such as DNA. DNA molecules are packaged into thread-like structures called chromosomes. Each chromosome is made up of DNA tightly coiled many times around proteins called histones that support its structure. DNA stands for deoxyribonucleic acid. Deoxyribose is a type of sugar that is used to help form the backbone of the DNA polymer and binds to nitrogenous bases. Transcription and translation. Transcription is the process of copying a segment of DNA into a segment of RNA. The pieces of DNA that are transcribed into an RNA molecule can encode different types of proteins and amino acids. Here I will be talking about mRNA, RNA polymerase, and uracil. mRNA is the form of RNA that is transferred to a ribosome in a sequence of bases. RNA polymerase is the enzyme responsible for the copying of a sequence of DNA into a sequence of RNA during transcription. Uracil is a compound found within living tissue as a base of RNA and DNA. Its place is taken by thymine. Next, I will talk about translation, which is the process of which the ribosomes in the cytoplasm or endoplasmic reticulum synthesize proteins after transcription in the nucleus. Some key components I will mention are tRNA, RNA, an anticodon, ribosomes, amino acids, proteins, codons, and stop codons. tRNA is a molecule that decodes messenger RNA sequences into proteins, which are nitrogenous compounds that consist of large molecules made of large chains of amino acids. Amino acids are organic compounds that contain amino and carboxyl functional groups. Key elements within amino acids are carbon, hydrogen, oxygen, and nitrogen, although other elements are found in side chains of amino acids. Another part to translation is rRNA. This is the type of RNA molecule just like tRNA and mRNA. rRNA helps cells form the part of the protein synthesizing organelle known as a ribosome and helps translate the information in messenger RNA into protein. A ribosome is a minute particle consisting of RNA and associated proteins found in the cytoplasm of a living cell. They bind messenger RNA and transfer RNA to synthesize polypeptides and proteins. Codons are sequences of 3 nucleotides which form a genetic sequence in a DNA or RNA molecule. Like codons, anticodons are sequences of 3 nucleotides which form a genetic code. But unlike a codon, the anticodon forms the genetic code in a transfer RNA molecule, corresponding to a complementary codon in the messenger RNA. Lastly, we have the stop codon. The stop codon consists of a genetic code within messenger RNA that signals to halt protein synthesis. Changes in DNA. Finally, we have the different changes or mutations in DNA, such as point mutation, substitution mutation, frameshift mutation, insertion mutation, and lastly, deletion mutation. Point mutation is a mutation which affects one or a few nucleotides in a gene sequence. Substitution mutation is a mutation where a base pair is replaced. Frameshift mutation is an insertion or deletion involving a number of base pairs that is not a multiple of 3, which disrupts the triplet reading frame of a DNA sequence. An insertion mutation involves the addition of genetic material. An insertion can be small or large. Lastly, we have the deletion mutation. This is where there is a mistake in the DNA replication process which removes nucleotides from the genome. This mutation can remove a single nucleotide or entire sequences of nucleotides. And that wraps up the video. Thank you.